So I'm delighted to be here, and I'm going to talk to you today about imaging research. And mostly I'm going to focus on imaging research that we're doing in our laboratory here at Yale. And so part of this is to educate you about the work that we're doing, educate you about the field broadly, but also, frankly, it's to help recruit talented young students like yourself to work in the laboratory here. So watch this with that in mind, and feel free to interrupt me at any point in time. And I'll try to save plenty of time for questions at the end as well. So we're interested in this construct of social perception in the lab. And if you have to define this, you're thinking about the initial stages of processing that allow you to accurately figure out or diagnose another person's intentions on the basis of their actions. So think about biological motion in terms of where they're looking, the ways in which they're walking, etc. How you use those cues to figure out someone's intentions, and also something about their psychological dispositions, even things about their personality. And I'd like to show this as an example. So what is this? You might have seen one of your other speakers present some of these. This is easy, not a trick question. A person walking, thank you. So you're able to figure that out based on a fairly impoverished stimulus. And what we've done is just place infrared lights on the major joints of a person and filmed them in the dark with a camera that was sensitive to infrared light. And so via these simple points of light, given the way they're moving, you're able to very, very rapidly discern that this is a person walking. So I'm interested in how the brain accomplishes that in typically developing people. And it turns out that it's been very interesting to think about how the brain might fail at that in people with autism. And so in addition to saying it's a person walking, are they guilty, proud, or embarrassed? It's a little harder, but speak up. Guilty, proud, thank you. So hopefully most of you see this person walking as proud. So they're conveying pride in the way in which they're walking, right? So again, this is a stimulus that's very impoverished in terms of what hits your retina, but nonetheless, your brain allows you, via your experiences and the representations that you've built up, and the actual realization of those representations in neurons in particular parts of your brain, to understand that this is a person and they're proud, right? So we're interested in how human beings accomplish this. And the argument behind all of this research is that human beings have evolved something we call the social brain, right? So the notion here is that by virtue of their unique demands for social information processing, we as human beings have built these modules for doing things like individuating others, perceiving agents and their actions, perceiving other people's emotional states, analyzing intentions and motivations, sharing attention and intentions, and then representing another person's perceptions and beliefs. And what I've done here is purposely put these in order in a way that I think they actually build upon one another. So very, very young infants are able to individuate others, perhaps from birth. Perhaps very shortly after birth, perceiving agents and their actions. Perceiving emotional states takes a little bit longer, but still something done within the first year of life. Analyzing intentions and motivations the first year to year and a half of life. You begin to develop this, and over the course of the first 3 to 4 years of life, you're using what you've built sort of in a bootstrap fashion from these early types of information processing to do things that we as adults do, representing another person's perceptions and their beliefs. So even we can figure out what other people are looking at, and we can make inferences about what they're looking at, make inferences about what they're thinking about by virtue of understanding their perception. And then we can begin to understand how the why of what people do. So we not only just see what they do, but we start to understand that there's a why underneath it. And that why is sometimes not based on reality as we know it. So they might think something else is true and therefore act upon that something else. And so as human beings, when you start to understand that other people are acting on the basis of what they believe, then that opens up a whole other world, whole other type of reasoning that you can do. So we're interested in how the social brain, what are the parts of the social brain that allow you to do each one of these, how do they work together in order to do the more complicated aspects of it, and then fundamentally how is that altered in a disorder like autism, which we view as a social information processing disorder. So to give you some names and locations of regions, I'll talk extensively about the superior temporal sulcus today. And this is a region along your temporal lobe, and you think just above your ear. I'll talk a lot about the right superior temporal sulcus today and the posterior part of it. So imagine a region just above your ear. I'll talk about the amygdala quite a bit, this deep limbic structure. I've brought it to the surface here to show you, where roughly it's located, but to think about that as being projected down into the brain, as it's a deep limbic structure. Orbital frontal cortex I'll talk about briefly in terms of its role in representing reward value, especially social reward, something that's very relevant to our understanding of autism. And I'll talk about a ventral region here on the surface of the temporal lobe, on the bottom surface of the temporal lobe, that's selectively involved in processing faces. We'll talk about its role in face perception deficits in autism. So as I've already revealed a bit, what I'm most interested in the context of autism is the qualitative impairments in social interaction. By now you've already learned that autism is diagnosed and identified by this triad of deficits as a syndrome. I see the qualitative impairments in social interaction as the key to understanding the entire disorder. I see the other two aspects of autism as emerging from these fundamental deficits in social information processing. And so that's why we're interested in things like the social brain, because we think that by understanding normal social brain development, it'll give us a fundamental insight into autism. So the two findings I want to show you that will sort of occupy or justify everything else I show you today. One is that individuals with autism fail to spontaneously look at the eyes. And this is something that anybody that's ever met a child with autism could have told you, but we as scientists wanted to quantify it. And this was one of the first studies that I was involved in as a scientist in autism. I had been using an eye tracker to study infants and the way in which infants look at faces. So I wanted to understand, just across typical development, when is it that infants start scanning the eyes and the core emotionally informative features of the face. And I wanted to understand this so that we could understand how infants represent emotions. And so I had the opportunity to see some patients with autism, and these were adults with autism. And I had been studying 3 to 7 week old infants, and I had seen that at about 5 to 7 weeks you start to see a lot of eye scanning in very young infants. They sort of go from looking at the periphery of the face to scanning the eyes, and they seem to be seeing the face as a face at that point. But these adults with autism, these were very high-functioning adults with autism, coming into the laboratory, and we put them in front of our infant eye tracker, and we were monitoring their eye movements, and they sort of fundamentally failed to look at the eyes. And as I mentioned, they were high-functioning, so these were people that could carry on a conversation. At the time, I had no clinical background, and so I really couldn't have told you that they were any different from, maybe a slightly awkward person on the street. But they, when presented with these faces, failed to look at the eyes. And you can see what they did. Here's the little red diamonds. It's a bit far away for you, but the little red diamonds indicate individual time points of a fixation plot. And so you see that this person stared at, she actually had an unusual shawl on here over her shoulder, and the person stared at that the whole time. This guy had an unusual feature to his eyebrow, and so this person with autism stared at that the whole time. She had a slightly, I suppose, unusual lip feature, again focused on that the whole time. And this is in contrast to our typically developing participants, which 201 tended to look at the eyes the vast majority of the time, and occasionally did the sort of triangular scan path every so often during looking at the eyes to get information from the rest of the core facial features, looking at the mouth a little bit, looking at the cheeks, etc. And so here we had a difference that was just obvious. It didn't need statistics to bear it out. It fit with the clinical impression, but now we had a quantitative phenotype. But we didn't know why these individuals failed to look at the eyes. And this is something that we've pursued quite vigorously through our imaging research, and I hope I can tell you about part of that story today. Then there was another finding that intrigued me, and this is a finding from Simon Baron-Cohen's lab. So Simon presented kids with autism, developmentally delayed kids, and typically developing children. The developmentally delayed kids didn't have autism, but their IQs were matched to the kids with autism who had some intellectual impairment. And then typically developing children. And he showed them Charlie here. This is Charlie. And he would say, which candy is Charlie looking at? and all the kids could say, in this case, the polos, and you would say, well, which candy does Charlie want, right? So now link the eye gaze with a mentalistic status, a want or a desire. The typically developing kids would say, well, the polos, because in this context, how else can you answer that question? You don't know what Charlie's favorite candy is, but the one he's looking at is probably an indication of the one he wants or the one he likes. The developmentally delayed kids with intellectual impairment but not autism could answer that question, so they were fine. But the kids with autism, even the higher functioning kids with autism, failed to answer that question, right? So they couldn't link together the mentalistic significance of this direction of gaze with what it means, right? So which candy does Charlie want? They could say which candy Charlie was looking at, but they couldn't make that leap. So what I want to do the rest of the time is tell you about research that we've done to follow up on these behavioral findings and similar behavioral findings. And I'm going to tell you first about our research efforts designed to just map out the social brain. So looking at typically developing adults, college students like yourself, and putting them in the magnet in a functional MRI machine and monitoring changes in blood oxygenation level and trying to figure out how do typically developing people process social information, and can we chart out the social brain. Then I'll talk about how that's disrupted in adults with autism, okay? And then I'll move from there into what I think is the most exciting stuff, and that is looking at the developing social brain, right? So autism is a disorder of development. And in order to understand it as a neurodevelopmental disorder, we have to study children longitudinally, and preferably very, very young children longitudinally, so we can really get at what is the primary pathophysiology as opposed to what are the effects of having autism and living in the world, right? So I'll try to make that distinction with some work. I already mentioned that most of what I'll be talking about today is functional magnetic resonance imaging as a technique. This is a technique that allows us to use an endogenous contrast that's present in the brain. The fact that when neurons fire, they use more oxygen and also more blood flows to the region of neural activity allows us to design pulse sequences using a large magnet that very safely and very non-invasively allow us to monitor a correlate of neural activity, okay? And so I can talk more about the limitations of that and what that means after showing you some of the science if you're interested. All right. So when you think about understanding other people and this thing that I've called social perception, one of the most basic aspects to it is figuring out when something in the environment is a biological agent versus an inanimate agent or a non-living thing, that those two things demand very different processes in order to understand their behavior, all right? So one of the first things we did was try to define a brain region that codes differently or responds differently to biological and non-biological motion. And the way we did this, we put people in the magnet and we showed them this display. So they're wearing virtual reality goggles, and we had this animated display. And every so often a person would walk, as you just saw, or what we called a robot would move, or what we're calling the disjointed mechanical figure. So the same parts and movements as the robot, but not conveying the gestalt of walking. And then we had something that's complex and nameable, but it's not biological, this movement of a grandfather clock. And we tried to control for as many different movement parameters as possible so that at the level of sort of hitting the retina, for example, this stimulus and this stimulus are incredibly similar, but they convey different meanings to you. One is something walking, and the other is a non-biological motion, this sort of random movement, okay? And what we found was that this brain region, this is an inflated brain here, showing you the temporal lobe coming down, frontal lobes, visual cortex. This is the posterior superior temporal sulcus. And what you're seeing is that this brain region is responding much more strongly to our two biological compared to non-biological stimuli. And so this was very exciting for us in the sense that now we had a brain region that was doing the work of differentiating biological from non-biological in the environment, right? Now, as neuroscientists, we get really excited when we see a dissociation. And the idea here is that it's not so impressive to us if every part of the brain does something, right? It's more impressive if we can localize function and say this part of the brain does this other part of the brain does something entirely different, right? And so this part of the brain called V5, which is just that's about a centimeter back behind the superior temporal sulcus, maybe 2 centimeters in most people, is responding much more strongly to all 4 types of stimuli. So it doesn't care whether it's biological or non-biological. And this is a very well-known motion processing region that all primates share that's involved in processing motion in general. So this is the part of the brain that allows you to see movement to begin with, right? But it doesn't care whether it's biological or non-biological. That's done by this region, which is slightly forward to this one. So imagine the processing stream goes into the retina, back into visual cortex, is passed to V5, it's recognized as motion, and then it's seen as biological or non-biological, right? So now we had a nice dissociation because we had no reason to believe that people with autism would be disrupted in terms of their processing of motion in general, but we had lots of reasons to believe they would be disrupted in processing biological motion, and that's a story I'll come back to in just a few minutes. So we had a brain region that we deemed involved in social perception because it's making this differentiation between biological and non-biological motion. But what we didn't know was whether it was sort of a dumb region that just did that and nothing else, or if it would be sensitive to the context conveyed by the motion, or its intention, right? And so I'm using intention and context relatively interchangeably here. So we wanted to know if it's sensitive to the intention conveyed, the goal is another word for that, then it would respond differently if we were to manipulate that aspect of the stimulus presentation. So what we did was put people in the magnet, and we showed them a different type of biological motion, this time eye movements, right? And so think about how important eye movements are to figuring out what other people are thinking about. You know, if you're looking at me, it's reasonable to guess that you're paying attention to me, for example, one of the most basic aspects of this. Or if you're looking at something else, you're paying attention to that. So what we did was put people in the magnet, and they're looking at a virtual reality figure, this female character. And every so often, a checkerboard would appear in her visual field. And sometimes she would look directly at that checkerboard. And what we were reasoning is that if you're watching somebody, and you know it's just sort of a plain background, and something appears that's interesting in their visual field, a valid prediction or a reasonable prediction is that they will look at that object that just appeared. Okay? And so our congruent condition did exactly that. Our incongruent condition required a little bit more of the brain to figure out, because now this checkerboard appears and it starts flashing, and she looks away. All right? And so what we were doing is borrowing from the Handbook of Infant Psychology here. Many studies of infants have been done where looking time is used as a measure of interest, right? So in this case, we weren't interested in what the subjects were doing with their eye movements, but what we were interested in is what their neurons were doing in terms of responding longer and stronger if something didn't meet their expectation, right? And when I talk about expectation here, I want to be clear that I'm not talking about the type of expectation that we commonly think about in terms of a folk psychological sense. You know, the expectation that you're going to go eat dinner after this, and if it's violated, then it requires additional processing, but it's a very cognitive type of expectation. I'm talking about a very basic implicit expectation about what somebody ought to do in a given context. And the reason I think of this as a very implicit type of expectation is that the subjects see hundreds of trials of this. So, and it's equally likely for it to be congruent or incongruent. So there's no right answer here. What we're doing is actually taking advantage of the types of statistical regularities that these neurons have seen from birth about what people tend to do in certain situations. And these neurons have essentially learned to respond differentially to these incongruent versus congruent situations in the service of figuring out other people's intentions. That's our argument. So, as you might guess, well, two things. One is that we were able to localize exactly where in the human brain people process eye movements. So if you wanted to know that and talk about it at a cocktail party, it's the right posterior superior temporal sulcus, about 35 to 40 millimeters from the anterior commissure, which is right about here. All right? So what I'm doing is slicing up the brain using imaging rather than a knife from 0 back to 60 millimeters from the front of the brain to the back. And what I'm showing you is yellow is the right hemisphere, and each one of these is a hemodynamic response, meaning that the brain is active in this location. And so what you're seeing is that in the right hemisphere STS, relative to the blue line, which is the left hemisphere, you've got recognition of eye movements in the brain, right? So we're very excited about this because on the one hand, we were seeing for the first time where in the human brain eye gaze processing occurs. But then we found that it actually mattered whether it was congruent or incongruent. So this opened up a whole new thought process for us. We had a brain region that's sensitive to biological versus non-biological motion. And now we've localized where in the brain typically developing people process eye movements. And now we've seen that it matters whether the movement is goal-directed or not. So this brain region, in terms of its role in social perception, is involved in actually representing other people's intentions, right? So we wanted to think about that a little more deeply. So we didn't believe it at first, actually, and there were some reasons why we thought we might have some attentional confounds, etc. And we thought, well, you know, it's not a very strong manipulation of intention, right? I mean, really? Do you have an expectation that somebody's going to look at something here, you know, just because it's the only other thing they've got to do in the world? You know, that's not very strong. So instead, we gave people an intention and an expectation, all right? So we put people in the magnet, and we had them watch Caitlin here. Caitlin's my lab manager. So Caitlin looked at the red cup, and she expressed positive regard. And if I had the sound turned on, you would hear Caitlin go, oh, and then she reaches for the red cup. So here in this situation, given the red cup and the green cup, Caitlin's expressed liking the red cup, and then her next action is to pick up the red cup, all right? Now here's another. So she likes the red one, but she picks up the green one. So this is very, very similar to the last study I showed you, but now we've sort of knocked our subjects over the head with, you know, Caitlin likes the red one, so your brain starts to make this implicit expectation that she's going to pick up the red one, and then we violate it. We wanted to make it a little bit more complicated and also control for visual attention. Whoops. So we presented Caitlin disliking the green one. She says, yuck. And then she picks up the red one. Well, in this case, it's actually the congruent thing to do to reach into the other side of the table. And that was important to control for spatial attention. And then she likes the green one. Dislikes the green one. Sorry, I got the conditions mixed up as I was describing them. But she picks it up, all right? So how often do you see somebody pick up the thing that they express that they dislike when they could just as easily pick up the other one, all right? So what we predicted was that in the incongruent cases, you would see additional activity in the superior temporal sulcus, in the right hemisphere, in the posterior section of it. And that's exactly what we saw. So we had a replication of our prior finding that this posterior STS region is involved in processing biological motion and it's sensitive to intentions, right? So that's the last part I'm going to show you that just involves typically developing people. Because at this point in the research program, we felt like we had a good enough understanding of this particular component of the social brain to start trying to understand something about autism, right? So what we did, given that study I showed you of Simon Baron-Cohen's, what would you predict would happen if I put some subjects with autism into the magnet and scanned them looking at Caitlin or that other study that I showed you with the avatar that looked towards the checkerboard or away from it? What would you predict? They wouldn't show a difference. Specifically where? Right, in the STS location, right? So they wouldn't differentiate the expected and the unexpected because presumably they're not forming those expectations and that brain region isn't supporting that formation of the expectation. So what we found, so we put them in the magnet and we showed them our incongruent versus congruent avatars. And what we found was that typically developing people in the right superior temporal sulcus differentiating congruent and congruent, just as we thought they would, and replicating the old studies. But in the individuals with autism, they failed to represent that intention. So they're responding equivalently to the incongruent and to the congruent. In a sense, it's as though their brain region is giving a closer representation of reality, because those two stimuli down to the pixel are actually identical in terms of how much movement they convey. So from the point of view of the retina, those are exactly the same stimulus, and we counterbalanced for side of presentation of the checkerboard, et cetera, to make sure of that. So many brain regions in the typically developing people respond perfectly equivalently. It's whether or not you form that expectation that drives that superior temporal sulcus activity. And the individuals with autism were failing to do that. So having just told you that as a group, they fail to form that expectation, they fail to show that differential activity, of their superior temporal sulcus lacks that differentiation. Now I'm going to tell you that there was a lot of individual variability in that. Okay, so the one thing, if you haven't been told already, that's absolutely true in autism, is that it's incredibly heterogeneous. Right? So even though we'll call two people, we'll say, they both have autism, the presentation of the autism can be very, very different. And one of the dimensions upon which it can vary is in the severity of the social dysfunction. So what we did was go in and look at the difference between incongruent and congruent in the superior temporal sulcus. And we said, okay, in this region, this is a measure of intactness of this brain region. Does it covary with social dysfunction? And we used as our measure of social dysfunction the ADI-R, the Autism Diagnostic Interview Revised. And there's a subcomponent of it called the Social Interaction Score. And this is one of the gold standard techniques that we use to diagnose autism, particularly for research purposes, so that we know that the people we're studying have autism. So all of these people met criteria for autism, but they still varied in terms of the severity of autism. And as it turned out, there was a very, very strong correlation between the severity of their autism and the measure of intactness of their superior temporal sulcus region. So if they had a superior temporal sulcus region that was more normal compared to the rest of the group, then they had fewer difficulties in social interaction. And what was exciting to us about this was that here we had a measure, a biological measure, that covaried with the severity of autism. So in a way, we started to wonder, could we use this measure to predict outcome, for example, in children with autism? Well, we don't know yet how severe it's going to be, but now do we have a measure that would allow us to start to be able to say, well, this child's going to have more dysfunction than this child based on the brain. We had a biomarker for the severity of autism. So again, one of the things that you'll learn about imaging research in autism is that it's very, very important to replicate. There are many, many, many results about the brain in autism that have failed to be replicated. And there are vast reasons for that. And so it's important to try to replicate. In this case, we replicated within our lab. I'd rather see this replicated across 2 labs. So I'm hoping that somebody will follow up this finding, although several people have found dysfunction in the superior temporal sulcus at this point. But we followed it up in our lab using this Caitlin task with the green and the red cups, etc. And what we found was that we could very nicely replicate our finding in typically developing participants. I apologize for the yellow on the white screen. This says yellow equals study 2, red equals study 1. So what you see is that the regions of activation are perfectly overlapping between 2 different studies done on 2 different magnets, right? And then we found that individuals with autism, again, fail to differentiate in their superior temporal sulcus the incongruent versus the congruent activity. So again, another indication that something is dysfunctional in this specific brain region. And then here's your typically developing results. But notice how big these standard error bars are. Okay, so there's lots of variability in this group still. So this time around, we had more information about who these subjects were. In particular, we had collected genetic material. So we knew their genome. And in particular, we had genotyped them for a particular allele, a particular gene called the serotonin transporter gene. And this gene is involved in regulating the amount of serotonin in the brain. And one of the old biological findings now, and controversial but for the most part replicated, is that individuals with autism have differences in terms of their amount of serotonin that they have circulating in their blood, and differences in serotonin synthesis in the brain. And so we thought, well, this is a reasonable gene to go after. In addition, the serotonin transporter gene has been linked to other components of the social brain, in particular the amygdala. So we thought, okay, we have this gene, we've mapped it in everybody. Let's see what happens in terms of our sample of individuals with autism. And what we found was that individuals that had the long configurations, they're different configurations. If you have a short allele on your serotonin transporter gene, then you have more amygdala activity in general, and you have higher levels of serotonin circulating in your brain and in your blood. So these individuals, the lows, with autism, now everybody here has autism in this bottom graph, but these are the individuals with the long. It seems to be a protective factor for exhibiting dysfunction in the right posterior superior temporal sulcus. So their social brain is more normal by virtue of having this gene. And then conversely, if they have a short allele, their social brain is more disrupted, right? So they're actually driving this effect. So here we have a gene linked to a particular, I think, important aspect of the social phenotype in autism, then linked to a brain region. So we can start to understand how, if you have autism, that autism is against a background of different genes from person to person. And we can begin to understand some of the variability in the autism phenotype by understanding other aspects, not necessarily the genes that cause autism, but the genes that are related to social functioning in general, and how those overlay on the genes that cause autism that gives you ultimately a variable phenotype, right? And that's what I think we've done in this particular study. And it's worth noting that it really doesn't matter what genotype you have if you're typically developing. Your superior temporal sulcus still differentiates incongruent from congruent. It only begins to matter if you have the autism overlaid on top of that genome, okay? That particular gene. All right, go ahead.
Does that also relate to behavioral measures like the ABI?
Thank you. In both our study and also a much larger study done by Kathy Lord, she found that she didn't have imaging data, but she looked at kids longitudinally, and the ones that ended up having the worst or the more difficult social phenotype had the short allele of the serotonin transporter gene. And then in our study as well, the lower ADI scores in terms of greater social deficits were in the kids that had the short allele, all right? But it doesn't covary with IQ. So it doesn't affect intellectual functioning. It seems to be specifically related to the social impairments. And one thing I didn't say about our prior imaging data and also Kathy's data in the serotonin transporter story is that it's not related to repetitive behaviors or language abilities. It seems to be specifically related to the social abilities, right? Thank you. So I want to leave the superior temporal sulcus for a second and take you to a different brain region, now one that's on the bottom surface of the brain, again in the temporal lobe. And I want to talk about this notion of why individuals with autism might fail to look at the eyes. And so this is particularly intriguing, I think, to clinicians in terms of, how do you interact with children with autism? There's some suggestion that sort of making individuals with autism look you in the eye, that's what this book is about, will tend to cause some anxiety or some painful sensations in the individuals with autism, that this is just overwhelming and disturbing. And so we wanted to know, well, we started with the finding that in this face area of the fusiform gyrus, there's hypoactivation, and that was a well-replicated finding. But the suggestion was, given the findings of differences in eye movements that I've already shown you, well, is it the case that you're just sort of scanning apples and oranges in that the individuals with autism simply aren't looking at the eyes? So it isn't that their fusiform gyrus is disrupted, it's just that they don't look at the eyes and they don't develop expertise for faces. And so their fusiform never develops that specialization for faces, so if it's disrupted, it's disrupted because of that, right? So what we did was actually put the subjects in the magnet and we showed them a face, which is a strong stimulus to elicit activity in the fusiform, and we monitored their eye movements, right? So most studies prior to this, not monitor their eye movements, we actually experimentally manipulated their eye movements. Most studies prior to this had either monitored the eye movements and noted a correlation between looking at the eyes and fusiform activity, or they had simply failed to control eye movements at all. All right? So we had a correlation that suggested that our hypothesis that by manipulating the scan paths we might see more normal function was true, but it was just a correlation at that point. So we experimentally manipulated eye movements, and we had several conditions. One was fixation. So you've got a crosshair that would appear, just a single crosshair, and it would kind of jump around, right around in the nose, the center of the face, okay? Then we had free viewing, and as the name implies, subjects were allowed to look in any way they wished. And this was very similar to all the studies that had been done prior to this. Then we had low looking at the eyes. So here we have sometimes the crosshair would land on the eyes, but it would also land on other features and off features entirely. Then we had medium looking at that, and then we had high. So by the time we got to high, we had about 56% of the time spent looking at the eyes, which is, we picked this range given what we knew about typically developing people, who tend to exhibit a range between 32% and 56%. Interestingly, and as a side note, that actually varies strongly with personality. In particular, if you're highly neurotic, you spend lots of time looking at other people's eyes. And if you're very low in the personality trait of neuroticism, you look very little at the eyes. And also, neuroticism co-varies with the serotonin transporter gene. So, one of the things, if you're a highly neurotic or anxious person, a doctor might prescribe a selective serotonin reuptake inhibitor, and that's how that works. It alters that gene's expression. So, back to the study. So what we predicted was that in individuals with autism, in the free viewing condition, they would fail to show activation in the fusiform gyrus. And that was a known thing already. But as we moved to more looking at the eyes, we would actually increase activation in their fusiform gyrus. In a way, this is an experimental demonstration of the frequent habit of clinicians to encourage eye contact in children with autism. It's one of the most basic things that you do in interacting with these kids in an intervention setting. And here we're trying to see how this might work in an experimental setting by putting them in the magnet, right? And so what we found was that just during free viewing, we replicated very nicely studies we had done, studies Bob Schultz had done, other people as well, that the fusiform gyrus, it's right here, was much more strongly activated in the typically developing people compared to the individuals with autism. Now, in addition, this brain region is called the amygdala, right? And so you see bilateral amygdala activation that's stronger for the typically developing people than for the individuals with autism. This is another finding that has been seen a few times during free viewing in studies of individuals with and without autism, all right? And the amygdala is actually thought to help direct your attention to salient things in the environment. Things that you don't understand, things that you understand but are very emotionally salient to you, all right? So the amygdala has long been of interest to people studying autism because it's a brain region that helps you sort of emotionally tag the topology of salience of things in the environment, typically leading you to look more at people's faces than to other things, right? Now, when we manipulated eye movements, though, during free viewing, you see this nice differentiation between individuals with autism, here coded in red squares, and blue circles for the typically developing individuals. So you see nice differentiation in the overall level of activation from the fusiform. When you make people that are typically developing fixate on the center of the nose, you actually drop their level of activity significantly, all right? And you make them look just like individuals with autism, and you bring individuals with autism slightly up by making them fixate on the nose, all right? So a significant jump already from just free viewing, where presumably they're not looking at the eyes much at all, to central fixation, just look at the center of the nose. Then as you move it to low eyes, you see that it's back to normal levels for typically developing people. And then after that, it really doesn't matter how much you make typically developing people look at the eyes. Their fusiform still responds. But the individuals with autism, you see a nice increase from low to medium, and then it levels off, and there's no difference between high and medium. So in essence, what we're showing here is that individuals with autism appear to have a functional fusiform gyrus. It's being masked by the behavior they exhibit, right? So they're not looking at the eyes. One of the very best things to drive the fusiform response is eyes. If you remove the eyes from a face and you present the face, the fusiform will still respond, but it will be a weaker response in typically developing individuals. And indeed, when you make individuals with autism look at the eyes, you see normal levels of fusiform gyrus activity. But how about the amygdala? So it appears that individuals with autism have an intact fusiform. It's being masked by their eye movements. This actually has implications for theories of where the fusiform comes from to begin with. So there's a huge debate within the world of psychology about whether or not the fusiform face area is involved in processing faces per se versus other categories of objects for which you have visual expertise. And one of the key findings cited by individuals that hold this sort of expertise point of view is that, well, individuals with autism don't have expertise for faces, and they lack a fusiform gyrus, and that's consistent with the theory. And what I'm saying is, well, actually, they have a fusiform gyrus that's responsive to faces, even though they lack expertise for faces, in any definition of the term. And they tend to do whole part space versus configural processing, et cetera. So this data is actually evidence against an expertise point of view and in favor of a more modular or a face processing region as a face processing region point of view. But the amygdala, well, the amygdala only responds in typically developing people when you don't make them look at the eyes. And at first we found that counterintuitive. We thought, well, we know the eyes, contain most of the emotional information in the face, especially a fearful face like the one we showed here. Why is it that when you make them look more at the eyes, you diminish activity? And it doesn't matter at all whether you make individuals with autism look at the eyes, right? Still, zero activity all the way across the board. So we couldn't recover amygdala activity, but we could recover fusiform activity. So again, showing a dissociation there. So the way we're interpreting this is that we think that this brain region is actually involved in distributing visual attention, eye movements to the eyes. So to answer the question, well, why don't individuals with autism look at the eyes? Well, I think it's primarily because of an amygdala dysfunction, okay? And this is consistent with what other people have theorized as well. But I think here we have experimental proof showing that you can recover activation in one brain region, but not in the amygdala. And the amygdala seems to only be involved in typically developing people when you leave them to their own devices, when the amygdala has a job to do, which is to direct eye movements. Otherwise, you're taking the amygdala out of the loop, and you see a zero activation in it. Okay? Okay. So everything I've told you about, excuse me, so far is focused on studying adult individuals with autism and typically developing adults, all right? So the problem with that is that everything we're seeing could as well be the result of having autism for many years, as opposed to any type of primary pathophysiology. And that's a big issue in the field of neuroimaging and one that we're all wrestling with. And the answer is to try to study younger and younger children. So I'm going to tell you about our efforts to do that. So I mentioned that the MRI machine uses this endogenous contrast, that it's non-invasive and perfectly safe. It is nonetheless very loud, very imposing, and not at all a pleasant experience. So you're getting into this big tube, and it makes lots of noise, and you're kind of slid back into the tube, and you're told to be perfectly still for the whole time. Not things that kids in general are good at, and particularly not the kids with autism. So we have to figure out ways to make this as attractive as possible and as manageable as possible for the kids. And so what we do is to take the imposing magnet and try to make it much more friendly. And one step that we take is to use a fake scanner. So this is called a mock scanner. And we build a mock scanner that has a lot of the same parts as the actual MRI scanner, but it doesn't have the magnet, so you can crawl all over it. There's no safety concerns, and you crawl inside it, etc. And then we put facades in front of the magnet, which are actually painted on fiberglass so that they can be brought into the real magnet. And I keep mentioning magnet, and indeed it's a huge magnet that's always on. So you can't walk into the room and have any type of metal anywhere, in your pockets, because it will fly out. And if there's a subject in the middle of the bore, the metal will be drawn very, very rapidly to the center of the bore and can actually hurt somebody very, very badly. So it's important that these are drawn on non-magnetic material, non-magnetic paints. And so we introduce the child with autism, a typically developing child, to Thomas the Tank Engine here, or the rocket ship, and this becomes a game. And so the game is to get inside of Thomas the Tank Engine or get inside of the rocket ship and show us how still you can be, play the statue game. And as you play the statue game, you progress through this process and you learn how to be still for extended periods of time. So we have a device that actually monitors head movement. This is a child in the magnet. This is a radio frequency head coil, which is used to take images of the brain. She's wearing glasses that allow her to see behind her head. And back here we have an LCD monitor that's playing her favorite movie. And then we have this little device here that's a little magnet that's being monitored in space. And this is a readout of how much she's moving. And this is in millimeter increments, so you can see some movements here from 0 to 10 millimeters. If she gets beyond 10 millimeters, then the computer program actually shuts off her favorite movie. And so therefore, she doesn't get the thing that she's enjoying watching. Right? And kids get this game very, very quickly. At first it's fun to make it start and stop, and then they want to be still so that they can watch their favorite movie. And so what we're able to do by doing this is actually scan kids all the way down to the age of 4 years while they're awake in the magnet, behaving and being still. All right? So that for us was a big conceptual, a big practical leap forward, and it allowed us to ask a lot more interesting science questions about the development of the social brain in individuals with autism. So one of the first things we did was go after this idea of whether or not individuals with autism differentiate biological from non-biological motion. So we showed the stimuli I was showing you earlier, but this time we showed it to very young children with autism. And indeed what we found was that in these kids, remember my plot before of the superior temporal sulcus, this is the exact same idea, going from the front of the brain to the back. Typically developing kids nicely differentiate biological from non-biological motion. Again, about 40 to 50 millimeters back into the brain, okay? Right hemisphere, biological motion in yellow, non-biological motion in blue. But the children with autism fail to make that differentiation all along their superior temporal sulcus, right? So we have a biological basis for the failure to perceive biological motion properly in these children with autism. This is showing you a summary activation. And again, showing you that what I'm trying to put forward is not a view that the entire brain is different or all aspects of information processing are different in autism. What I'm trying to convey is that social information processing and social information processing regions are affected in autism specifically. Right? And what this is showing is that the posterior STS is differentiating typical development from autism. It's dysfunctional in autism. But again, this our area that we talked about before, V5, it's behaving perfectly normal in autism. So the prediction would be that individuals with autism would perceive motion just fine. But when called upon to make the differentiation between biological and non-biological motion, those individuals have problems doing that. And indeed, that's exactly what you see behaviorally. So here we have, again, evidence for dysfunction in the specific brain region. We followed this up, as I've tried to hit you over the head with. It's always very, very important to replicate every finding in the field of autism. We followed this up with point light walkers. And this was stimuli that were borrowed from Ami Klen and Warren Jones and colleagues that convey these sort of complex, very rich social actions. This is the now famous patty cake video conveyed through point light displays. Okay? And what Ami and colleagues found was that when you compared non-biological to biological motion, very, very young infants with autism failed to orient preferentially to the biological versus the non-biological motion. And then others have found that children with autism fail to differentiate biological and non-biological motion, older kids. So we showed biological motion versus a scrambled control. Okay? And so what we found, in this case we had 3 groups of subjects, all right? Jamie, we go until 5, right?
Yep.
Okay, excellent. So what we found was that in 3 groups of subjects is something that I think is very, very interesting. Neurotypical individuals, yellow and orange here, are showing you where biological is greater than non-biological motion. Okay, so you see that in regions in the left hemisphere that are typically thought to be involved in language production and naming things, and language understanding and semantic understanding here, so Broca's area, Wernicke's area, motion processing regions, V5 is right there, and the superior temporal sulcus, all strongly more active to biological than non-biological motion, right? And then you can see here in the right superior temporal sulcus, much stronger activity to biological versus non-biological motion, also in the bilateral fusiform gyri, okay? Now, then we had, these are neurotypical kids, so these are just typically developing 5 to 11-year-olds. Then we had unaffected siblings of children with autism. So these are kids that do not have autism, but they're related, they're a sibling of a child that does have autism. And those were our third group, the kids with autism, right? So what we were thinking when we first started this study is that these would be an excellent control group for kids with autism because they're growing up in families where, you know, they're being affected by autism, but they don't have the phenotype, they don't have the disorder, okay? So what we found, though, was that the unaffected siblings actually showed decreased activity in superior temporal sulcus in this parietal cortical region that likely corresponds to Wernicke's area, and also in this frontal region that corresponds to Broca's area, all right? So they were showing evidence of a brain phenotype that was actually much more consistent with the brain phenotype that you see in autism than in the typically developing kids. And that was striking to us because what it suggested was that we had here a picture of a vulnerability for developing autism, a brain vulnerability or a brain phenotype, or what people talk about in terms of endophenotypes, something that doesn't give rise to the full-blown disorder, but is still a trace of the vulnerability for developing that disorder on the basis of your biological constitution, okay? So again, they don't have autism, so nothing you're seeing here is the result of autism. There's no differences in expertise, etc. Their brain is showing a differentiation based on a different vulnerability to autism, right? And so follow this up. We also looked at the amygdala. So you see that individuals with autism completely lack amygdala activation to these stimuli. Unaffected siblings are showing only unilateral amygdala activation. Typically developing people have it in both hemispheres. And also, this was very interesting to us, this activation here, because it corresponds to reward circuitry in the brain. So here you had regions that are known to be involved in very simple rewards, like delivery of cocaine, actually, in rats, and when you get paid a little bit of money or win a hand at poker. These are the regions that come online. When you see a beautiful person, these are the regions that come online. And what they're doing is coming online in these typically developing people for biological greater than non-biological motion. But they're not coming online in the unaffected siblings or in the individuals with autism. So it's as though they're not finding these stimuli rewarding in any way, shape, form, or fashion. Now, we don't have behavioral data. We didn't ask the subjects, did you find those rewarding? But using this imaging technique, we were able to see brain regions that are consistent with those interpretations, differentiating these groups and failing to differentiate these two groups. And then finally, you know, it's equally important to see a brain region that works. So this is V5. We had a little bit more motion in our biological compared to our non-biological stimuli. And indeed, V5 revealed this. So it's equivalently active in all 3 groups, right? So what I want to convey is that autism is not a disorder of all aspects of information processing. It appears to be a disorder that most strongly affects social information processing. And I think that's what we're seeing here. So one of the things that we're getting into now, given this data concerning a vulnerability for developing autism, but the lack of the actual full-blown phenotype, we're deeply interested now in taking this even younger. So what we want to do now is look at infant siblings of people with autism. So these are kids that, you know, they're newborns, they come into the lab and they're seen longitudinally over time, and they're seen because they're at increased risk given the heritability of autism. So it runs in families. We know that these individuals, if they have a first-degree relative with autism, are more likely to develop autism. but they haven't yet. And we compare those to kids that are at normal risk for developing autism, and we watch them over development. And so what we want to do next is now not only watch them behaviorally, but start to try to understand how their brain is functioning throughout this period of development to see who could we predict which individuals would develop autism versus those that don't. We also want to start to try to intervene, because one of the strongest tests of our hypothesis about this being a disorder of social brain function is to have a social information processing intervention that would happen very, very early in life before the identification of the autism, and then see if we can set a number of kids on a different trajectory. So we watch how their brain actually alters as a result of having that sort of enhancement in social information processing delivered to them very, very early on. And we're talking about, playing with the children in specific ways, in ways designed to actually improve social brain functioning, and then monitoring the outcome as a measure of brain function. Okay? So what's important in order to be able to do this is to have techniques that allow us to look at brain function in very, very young infants. If Jamie hasn't spoken already, he'll talk extensively about evoked response potentials, which are one really wonderful way of looking at brain function, even in very, very young infants. Another way is to actually use MRI and functional MRI, which I've been talking about the entire time. It has some limitations, though, for scanning infants. They have to be asleep while you're scanning them because, again, it's a noisy, somewhat scary environment. And another way to actually image brain function is using infrared light. So if any of you have ever been to the doctor and had your pulse taken with a pulse oximeter, the notion is that they shine infrared light through your finger, and that infrared light bends differently and responds differently depending on how much oxygen is in the blood. So it's infrared light will pass right through skin and to some degree bone, but it's stopped by different properties of the blood and therefore reflects differently. So you can measure oxygen. So the principle here is that infants actually have quite thin skulls, so optical imaging works quite well with them. And so talk for a minute about near-infrared spectroscopy. So one of the things that we're doing is using this type of technique to begin to scan very, very young infants. And with some success, this is Liam, who's a child of one of the research scientists in the lab. And Liam has a hat on that has transmission and reception optodes. So these are little optodes, and they're connected to fiber optic cables that go back into a laser. And the laser generates near-infrared light, which is perfectly safe to tissue. You're getting a lot of near-infrared light now, actually. When you walk outside in the sunlight, you get lots of infrared light actually passing through your skull. And so Liam is having, on the blue, in the red here, light is being emitted, and it's traveling through the skull in a diffuse pattern, but it tends to sort of take a banana route most strongly. And it's reflected off of the cortical surface and back to this little blue one here, which is a receiver. And we measure the properties of that light as it's transmitted and received, and we can make inferences about underlying neural activity, particularly via oxygen levels. So it's the same exact measure as fMRI, but done with light instead of a loud magnet, right? So why don't we do this instead of fMRI all the time? Well, you're limited very, very clearly in terms of how deep you can go. So for example, you can't study the amygdala with this technique, unfortunately, but you can certainly study the superior temporal sulcus or the fusiform gyrus, both of which are surface features on the cortex rather than deep brain structures. So we've been doing this. This is showing you the number of optodes that we have. In this case, in this study, we were wrapping them around the back of the head, and these optodes here were hitting the superior temporal sulcus. And lo and behold, when we looked at our fMRI data from that study involving Caitlin reaching for the red and green cups, here's the fMRI activation. And from this recording optode, what we found was a stronger response to the incongruent versus the congruent, so solid versus dashed line. So we're excited about this in terms of being able to take into studying infants something very somewhat high level in terms of social perception and social cognition. So we think that infants, based on behavioral work, can make these sort of incongruent versus congruent intention distinctions. And now we can study how their brain is doing that in this particular region, and then we can begin to study the kids that are at high risk for developing autism versus the kids that are at low risk for developing autism. So if you were in this class next year, I would hope to be showing you data that might differentiate those two groups of individuals, and therefore give us some measure of prediction and also hopefully some measure of response to treatment in the individuals that are at low and high risk for developing autism. The last thing I want to show you is something that particularly those of you that are going to be working with the low-functioning kids with autism at Benhaven are going to come up against, but all individuals with autism tend to have a tendency towards emotion dysregulation. It's something that's very challenging for children with autism to learn, because one of the primary means by which you learn emotion regulation is through social interactions with others. So if you're dysfunctional in terms of your social interactions, then you're far behind in your ability to actually regulate emotion just from the starting point. So we've been developing ways to study emotion regulation, right? So again, it's not a primary characteristic of autism. Many people have emotion dysregulation, but it tends to be more true of individuals with autism than individuals without autism. And it's something that's very troubling to parents. So if you're the parent of a child with autism and you go into a grocery store, a lot of times it feels like it's only a matter of time before there's a meltdown, and it usually ends in you leaving the grocery store prior to being able to get what you came for. So, with people looking at you in a funny way. So we developed this task to study emotion regulation in young children in the magnet. So the kids are playing a game, all right? And they see a series of green and red squares, and inside of those green and red squares are just common objects, and we also show faces. Not central to the point I'm going to make, but the faces are there. So when they see a red one, their job is to not press the button. And when they see a green one, their job is to press a button. So this is called a go, no-go task. It's a very typical kind of tasks for people who are studying cognitive inhibition, right? But what makes it an emotion regulation task is that before they play the game, they're told, all right, if you win, you're going to get a picture of your brain, and you're going to be able to put your name on the wall of fame, and I want you to go over to the table where you see all those really cool toys, pick one out, because you're going to be able to take it home, but only if you win. If you lose, you're out of luck, all right? So there's lots of demand already. They pick out a toy, they play with it for a few minutes, they show it to their parents, and then we take it away, and we put it on the table, and we say, well, if you win, you get to leave with this. And then we put them in the magnet, and they're playing this game. But unbeknownst to them, there are 3 blocks to this game. At the beginning of the game, everybody wins, right? So it's pretty easy. You're winning, and you're being told, by this little guy here, that you're winning points, right? He comes up every so often. There's a little thermometer that shows you in real time how many points you've got and how close you are to your goal, right? We even put a little picture of the toy they want on top of that thermometer. But then in the middle block, everybody loses. So this is like a game at the Durham Fair. No matter how good you are, you're going to lose most of the time. So every kid at this point is losing. And now they've got to emotionally regulate, because they're going to lose, from their perspective, something that's very, very important to them, right? So we bring kids almost to the point of sort of meltdown by modifying the difficulty of this task. And because we can modify in real time how fast the task is going, how hard it is, we can make everybody behave or perform at the same level, which is really important in interpreting brain activity, right? So everybody's losing to the same degree. They almost lose all of their points. And then in the third block, everybody wins, because the first rule of child psychology is that everyone leaves the lab a winner. So, but they don't know this. And then they have to inhibit the opposite of a negative emotion, which is, I'm winning, I'm getting what I want. And oftentimes in life, that's also something that you have to learn to emotionally regulate, sort of being a sore winner when you're getting what you want and not sort of taking your mind off of the task. So if you play sports, if you're up 3 goals in a soccer game and you stop playing defense, you've got to kind of emotionally regulate even though you're winning, all right? So we can study regulation by subtracting this block when now a strong negative mood has been induced by the losing block from this block, which is winning and presumably doesn't require any emotion regulation or not as much. And we can then identify brain regions that are involved. And just to show you that the task actually worked, when the kids got out of the magnet, we asked them, well, what were you thinking? And so this child, age 10, I thought I was going to lose all my points. I was very worried, so showing that the block 2 actually did work. It was sad when I was losing, but I knew I would really win. And so that's actually an example of an emotion regulation strategy where you reframe something to be not as bad. When I was losing, I pretended that all the bad faces were really happy. That's actually a very sophisticated emotion regulation strategy. So, if you're giving a talk and it's going poorly, you pretend everybody's clapping for you. So, sophisticated little 5 to 11-year-olds. And what we found was that when we looked inside of brain regions that are involved in emotion regulation, one of them is the anterior cingulate. So this is the very center of the brain. This is the corpus callosum. And above that is a strip of cortex called the anterior cingulate cortex, right? And in regions that are at the top of that cortex here, called the dorsal anterior cingulate, those are known to be involved in higher level strategies for emotion regulation. Things like reframing or pretending it isn't that bad or thinking about something else. Things that demand some cognitive ability to be able to do while you're under a lot of stress. But down here in ventral anterior cingulate cortex, this is a brain region that's known to be involved in the experience of negative emotion and in very kind of primitive strategies for emotion regulation. And what we found was that the dorsal anterior cingulate, its response during this block, nicely followed age. So as children grew older, their dorsal anterior cingulate became more online in block 3 versus block 1. Their ventral anterior cingulate went down in activity. So these kids are older and have less activation in this ventral region. So it's showing you dynamically in the brain over this sort of half decade of development how the brain normally begins to use one brain region versus another for emotion regulation. And all of us know this, in interacting with kids, that older kids are better in general at regulating emotion. But what this gives us is a sense of what the normative profile is for the development of emotion regulation. And then a insight into, as we're looking at this in autism, how that deviates in kids with autism. And therefore, potentially a place where we could go in and intervene, a target of intervention. So, however, we all know that there are some people who never really get emotion regulation. Hopefully none of them are your roommates. But there are some people for whom emotion regulation is just really difficult. They tend to be people who are very high in neuroticism, and they tend to be people who experience a lot of negative affect and a lot of fear. And in children, we call that a negative or a fearful temperament. So we measured temperament. So now this is irrespective of age. When you look at fear scores, so higher scores mean children who are more fearful. These are kids that are extremely shy, that tend to be afraid of everything new, et cetera, et cetera. These kids had lower dorsal anterior cingulate activity and higher ventral anterior cingulate activity. So what this was giving us as a measure is a biological brain measure of activity that relates to aspects of temperament. And temperament becomes personality as children develop. And so what we're seeing here is the beginnings of differences in what we think will be long-term outcomes in terms of how these children emotionally regulate. So now as we move this into the world of autism, we could begin to perhaps diagnose those kids that are particularly going to have particular trouble in developing emotion regulation strategies. And those could become the kids that receive specific interventions aimed at increasing emotion regulation strategies, which we don't think will affect their autism per se, but will certainly make it easier for other individuals to work on the characteristics of autism versus having to deal with those emotion regulation deficits throughout their life. And so, let's see. I think that's all I want to say. And since we're videotaping this, I'll put up the financial disclosures. We're very fortunate to have lots of funding from outside agencies and to do things like support undergraduate projects if I've managed to interest you today. And that's it.